Good morning, everyone. So my name is Didier Le Sueur, as my colleague told you. So this morning is a nice opportunity to share with you some results what we have got, not in Asia, but in Africa, because I spent 15 years of my life in Africa, working with African institutes. But I mean that wherever it is, the topics are relevant. In Asia and Africa, we are facing the same problem with soil fertility, and we are willing the same interest of promoting biofertilizers and so on. So today, the outline of my presentation will be, first of all, what is biofertilizers? I'm sorry I was not able to attend the previous session because I just came back from Europe yesterday morning, and I was pretty busy yesterday. But I will, after the definition of biofertilizers, I will show you briefly how microorganisms can contribute to improve crop yields. And I will talk about production of biofertilizers, because production is very important if we want to scale up and if we want to be able to produce very nice and very effective biofertilizers. I will talk about how to inoculate biofertilizers, because you will see that according to the plants, the protocol for inoculation can be different. And we will talk about a project that I was coordinating during 3 years in East Africa called Compro, funded by Bill and Melinda Gates Foundations, to promote biofertilizers in African agricultures. I will talk about quality of biofertilizers. This is something very important. That's why in this room, a couple of months ago, we organized a regional training involving people from Myanmar, Cambodia, Vietnam, Thailand, and funded by Crawford Foundations. And we organized it with Deakin University in Melbourne. And I will show you some results what we have got from the field with after inoculation of biofertilizers, and I will go through some list of conclusions and perspectives. So, what is biofertilizers? So you can see on this photo, very nice products, good packing, they look very attractive, they look very interesting, but unfortunately, many of them, they are totally useless. So, but what is biofertilizers? So you have several definitions, but I think the best one is the one defined by VSA 2003. A biofertilizer is a substance which contains living microorganisms which, when applied to seed, plant surfaces, leaves, roots, and soil, are able to colonize the rhizosphere of the interior of the plant. So in this case, we talk about endophyte, and to promote plant growth by several mechanisms, which final objective to improve plant nutrition, plant growth, etc., etc. So according to this definition, for the rest of my presentation, when I will talk about biofertilizers, please keep in mind this definition. Sometimes we talk about biofertilizers, sometimes we talk about bioformulations, sometimes we talk about other terms, but they are all meaning the same. And I give you a list of references of publications talking about, they are all review papers talking about biofertilizers. If you need to get them, just let me know. I have PDF of all these references. So how do biofertilizers can promote plant growth? So on this slide you can see many systems, agricultural systems, natural systems, and you can see that for every system you always have the same kind of microorganisms. For example, some biotic microorganisms here. So here, but you have it also in natural system. Same for free living microorganisms here, and you can get them here. So it means is whatever is the systems, you have always the same kind of microorganisms and the same kind of associations with plants. All how these microorganisms can contribute to improve plant growth. So you have several mechanisms. For example, let's talk about symbiotic microorganisms. So you can get mycorrhiza, they are fungus, as you know, and mycorrhiza used to improve phosphorus nutrition for plants. But we have also what we call where are they? You can see all the symbiotic microorganisms. They can form nodules on legumes, and they can fix atmospheric nitrogen, and they can provide nitrogen to the plants. But we also have no symbiotic microorganisms, such as, for example, P-solubilizing bacteria or P-solubilizing microorganisms. Phosphorus is widely present in the soils, but unfortunately it's not available for the plants for several reasons. So these microorganisms, they can solubilize phosphorus, and then they can make it available for the plants. And when we know that, for example, phosphorus resource is very limited, because we think that maybe in 20, 30 years, the natural resource of phosphorus will be completely exhausted, it makes a lot of sense to put efforts on P-solubilizing microorganisms to take advantage of what is naturally existing in the soil. And you have also microorganisms capable to produce phytohormones, other kind of molecules improving plant growth, but also to remove some plant diseases, pathogen agents. So they can play also an important role. And you have everything about biological control, but we don't have too much time today to talk about it. So just some people were thinking that because, of course, year after year we come up with new varieties of plants coming from breeding programs and whatever. So some people were thinking that maybe the previous cultivars were more responding to mycorrhizal inoculation than the new ones. So this review paper published in 2012 showed that it's the opposite. I mean, new cultivars were more mycorrhizal responsive than the ancestral genotype. So it means that the problem what we have been facing in the field when you want to inoculate plants with mycorrhiza is not the consequence of new cultivars, but it's a consequence of other systems. So just to show you that how it's difficult in the field to get positive impact of after inoculation of plants with mycorrhiza. I will show you more data later. So this is again about phosphorus. So I was telling you why it is important to put efforts on phosphorus. And you can see the key role played by microbial communities in the cycle of phosphorus at the field scale. And this is why it's very important to take them into account. So now it's time to talk about inoculations, methodology to practice inoculations, and which factor do we have to take into account. So what do we have to inoculate? Because this is the starting point. You need to know what do you want to inoculate, and you need to start from something effective. So once you have something to inoculate, which kind of formulation do we have to use? So I will show you the importance. And when do we have to inoculate? And we have technologies, and I will share. I'm going to share with you some data. So just to show you, let's start with the rhizobia legume symbiosis. So you can see here, sometimes you can get very different nodules. You can see here, they are all nodules coming from the same plant. All the strains also of rhizobia, some are used to acidify the media, the others can alkalinize the media, and you can see a rhizobia. How does it look? So when you have to elaborate a biofertilizers, you used to start from nodules, okay? Once you have the nodules, you isolate the rhizobia contained in the nodules. You need to select the effective strains by doing greenhouse testing. And after that, you move to the field and you want to confirm the results obtained under controlled conditions, because in the field is where you need to get impact of biofertilizers. So let's start with isolation. So this is the material that you start from, nodules, collected, for example, in the field or from greenhouse testing. Trapping, for example, if you start from soil, you need to trap a rhizobia. So you have to disinfect your nodules. You crush when they are clean. You streak on your culture media. You can make serial dilutions because you won't get only one kind of rhizobia, you may get several kinds. So by doing dilution, you increase your chance to come up with several kinds of rhizobia. When you have pure cultures, after incubators, you can put on agar media, and at least now you have your pure strains. Once you have your collection of strains, you need to know which ones are very effective. So you need to, from your collection, you start liquid culture, what we call broth, and after that you can carry out a test, inoculation test. So you have many devices to do it. You can do it in tubes, as I was doing when I was in Africa, but you can do it in patches, for example. It's pretty easy. So whatever is the system, the objective is to assess the amount of nitrogen fixed by the plants in symbiosis with the plants in symbiosis with the microorganisms, and to see the impact on the growth. So now it's about formulation. What do I mean by formulation? I mean, you cannot go to the field with liquid culture because we know that it's a big constraint. So you need to fix your bacteria in a carrier. This carrier will be able to keep the bacteria alive until you inoculate the microorganisms. So in this case, it requests a very appropriate carrier. It requests also that this carrier needs to be sterile. So you need to use good technology to make the sterilization effective and complete, and I will show you the consequences if the sterilization is not done correctly. And it's supposed to be easy to carry, to store. Storage is very important because, I mean, if you produce, for example, at LDD your products and you want to go to south, for maybe it will request some house by car, by road, and you are not sure about how it will be stored, you can strongly damage your products, and the end users, such as farmers, like farmers who are here today, they won't be able to get good results if the product has been damaged during the transport. So just to show you some examples of formulations, you have the very easy one, liquid inoculant, which is easy to prepare and to use, but we have a lot of constraints in terms of storage, as I told you, but also transport. Basically, people use peat because peat has the advantage to be a very good carrier, easy to use, easy to store, easy to carry, but it requests a very, very good sterilization. If your peat is not well sterilized, you will get a lot of contaminants, and at the end of the day, you won't get any good results in the field because you will get only contaminants in your products. And also something that we try to promote in Africa is alginate beads. Very easy to use, to store, to carry, but it requires some facility if you want to produce them, and I will show you some slides. And also the main constraint is you need to dissolve the beads with a phosphate buffer before application. So let's talk about alginate beads. You can get an example of alginate, fresh alginate beads. So all the protocols are available, and you can try to combine, for example, rhizobia and mycorrhiza on alginate beads, and I'm going to show you some results. So on this slide, I tried just to show you the several steps for the production of alginate beads. On your left, this is the mixture of alginate and broth, equal volumes. With a peristaltic pump, you just get your mixture here, go through the pump, and you can drop, as you can see here, in chloride calcium solution. And when each drops forms one bead. So this is fresh beads, and you can dry them, so you reduce the volumes, and at the end you have a full box of dry beads, and you can inoculate a lot of plants. So in terms of protocol, it's exactly the same. You have to prepare a solution of alginate 5% with distilled water, mix equal volume with the bacterial culture. Dropping system in chlorocalcium solutions, 0.1, 0.2 molar. You check. Once you have your beads, don't forget to make rinsage, because calcium chloride is used to kill bacteria. You rinse, you dry, you store, ready to use. So just, this is a smaller scale. When I was in Senegal, we set up this very simple device, and at the end we have got fresh alginate beads, you can see here. So the objective for us was to see if it was possible to include rhizobia, but also mycorrhiza, AMF. So what we did is we started from liquid culture and small pieces of roots infected with Glomus enteradices, and we mixed both with spit, and we ran some tests under greenhouse condition to compare mycorrhiza alone, rhizobia alone, beads with both microorganisms on both plants, 2 acacia species. And here you can see that you have a list of all the treatments at the bottom of the slides. But basically, you can see the names, but it doesn't matter. You can see that we have got good results with this technology. Now the idea is to scale it up, and to see if it can work at the bigger scale. But the project ran at the end, and we didn't get enough time to do it. I was talking about the carrier just to show you. I mentioned peat has a very good carrier, but we also try some vegetable powders, and you can see that with peat we store the liquid inoculants at 4 degrees or 28 degrees, and you can see that this number of variable cells didn't change after 28 days of storage. If you check with the vegetable powder, same conditions, you can see that the number of cells decreased after 28 days of storage. Just to show you the importance of making a good choice for the carrier. Just to illustrate a little bit what I was talking about. So now about methodology to use for inoculating your plants. We published 3 papers. They are all available, so if you need them, just let me know. just to show you how the methodology for inoculation can be different according to the species of legumes you want to inoculate. So basically, we identify many treatments. Inoculation with dissolved adenal beads, coating the seeds, pre-germinated seeds, with gum arabic, sticking agent, and liquid culture. Inoculation with liquid culture around the root collar of seedlings just after sowing. Inoculation with liquid culture, but 1 week after transplanting the seedlings. And inoculation with no dissolved vaginal beads containing rhizobia on control. And what happened is, for example, for Caliandra, Calotersus is a 3 nitrogen fixing species, very useful in agroforestry. It has been widely introduced in Vietnam and in Indonesia, for example. And we discovered that inoculation was optimal when it was done with liquid culture around the root collar of seedlings just after transplanting. For Acacia mangium, I believe that many of you know that. For example, in Vietnam, you have huge plantations of Acacia mangium, of course, fast-growing Australian acacia, but also Leucana leucocephala, very important nitrogen-fixing trees for agroforestry. We discovered that for Acacia mangium, the inoculation was optimized if you do it with liquid culture around the root collar of seedlings 1 week after transplanting. On the other hand, for Leucana leucocephala, is when you did coating of seed pre-germinated with gum arabic and liquid culture. And the last one, they are African acacia, native acacia, with very deep root systems. We showed that if you want to optimize your inoculation, you need to do it with dissolved agenate beads. Just to show you that according to the species, you have to adapt the methodology to do inoculations. So, what is really the potential for biofertilizer in tropical agricultures? We have some success story, for example, rhizobia and soybean, but they are very limited. Why is it limited? What is the problem, and so on? So, for rhizobia and soybean, it's a very long story, very nice story, because now you can see that, for example, in countries such as Brazil, Argentina, the yields is about 4 tons of soybean per hectare by using this kind of commercial inoculants. Containing, for example, if you go to US and you check, you discuss with farmers, I can tell you that most of them, they will tell you that they use these commercial inoculants containing strains of bradyrhizobia, but also what we call knot factors. Knot factors are, during the formation of nodules, for root nodules, you have several steps, and you have new molecules playing very important roles. So some people, they thought of fact that if you mix rhizobia with some key knot factors, you will improve nodulation and it will automatically impact on the soybean yields. And it's exactly what is happening in the field. And even when these products are expensive, I can tell you that all the private company manufacturing these inoculants are making a lot of money. So it's for America. What is going on in Asia? So far, I think very few things. In Africa, the only success story that I've heard about is Zimbabwe, where farmers, despite the very critical situation here, economical situations, farmers are still asking for inoculant when they want to grow soybean. Otherwise, we have almost nothing. And it's something that's very, very interesting and also very, how can I say, disturbing, because a lot of people are trying to promote biofertilizers. and the results are not so good. So just now is to talk about this project that I was coordinating during 3 years, called Compro. So the full title was Evaluation and Scaling Up New Chemicals and Biological Commercial Products for Improving and Sustaining Crop Yields in Selected Agroecological Zones in Sub-Saharan Africa. So project funded by Bill and Melinda Gates Foundations. So the idea was to say, the foundation was always asked by private companies, okay, I have fantastic products, magic products. You need to promote it, and you will see your farmers in Africa will get happy. But they didn't have the capacity to assess all these products. So they came to us when I was working at the Tropical Soil Biology Fertility Institute of SIAT in Nairobi. And they asked us, okay guys, you are scientists, so try to run field testing, try to see which one are effective, just dispose the useless one, and try to scale up, test in the field, scale up good products, and we will see after 3 years what you will be able to come up with. So we worked in 3 countries, Kenya, Ethiopia, and Nigeria. So the objective, one of the final objectives were to assist farmers, because if one farmer decides to spend some money to buy biofertilizers, he cannot get disappointed because the product is completely useless. So that was the objective. And the objective was also to move forward with a scientific approach and to start to work on commercial products. So we listed 5 objectives. The objective was to describe a baseline information about in the regions where the activities were supposed to be carried out. And after that, in terms of activities, we started by screening new products. So we got products coming from all around the world. I mean, yes, from Australia to US, Europe, Asia. We wanted to get what was available on the market. We try also to understand the mechanisms involved, and we try also to see how these products, specifically the good ones, could be effective on the local markets. And the last one was about management. So, okay, this is something very important, and I want to point out this morning is quality of the commercial inoculants, and you will see at the end of the presentation why it's so important. So why do we have to look at the quality of the products? Because, first of all, you have plenty of products on the market. You have almost no information about these products. Are they effective? Are they ineffective? Which kind of microorganisms they contain? Are you sure that they won't damage your crops? You don't know. You have almost no information. So when you ask to the companies, they are not able to come up with scientific results. They will show you photos where you will see one row of very poor plants, and you will get one row with very nice plants, and they will tell you this one is where we apply the products. But when you ask about scientific devices, how did you do it? Who did you do it? You won't get any answers. So that's why it was very important. And something also very key is to be sure that you won't put under danger farmers, because you will see that unfortunately, some of these products can contain human pathogens. So that's a very important concern. And the last thing is the absence of regulations. Few countries set up regulations, but most of them, they don't have any regulations. So it means all these companies are free to produce whatever they want. So yes, it's just to repeat what I was saying, is to prevent the proliferation of underperforming products, to add value to effective products, to help for promoting utilization of biofertilizers, and also to show at the top level that with no regulation we won't be able to optimize the promotion of biofertilizers. Okay, so as I was telling you, 65 products coming from all around the world, several categories of products, so biofertilizers with rhizobia, but also with free-living and fixing bacteria, some with phosphate solubilizing bacteria, and some with biological control such as Pseudomonas and so on. Just photos showing some of them. So we started with by contacting the companies and ask them for information. You can see that for some we didn't get any answers, and we distinguished several categories of products. 39 products with rhizobia, 7 only with free-living and fixing bacteria, and 16 with PSB biocontrol and so on, just to show you the breakdown. So in terms of methodology, we started from the products. We made serial dilutions, and for the good dilutions, we always try to cultivate microorganisms on selective and non-selective media, just to take everything into account. So just to show you an example of kind of cultures, what we came up with. So from each product, we selected the strands, and we store them in glycerol at minus 80 degrees. And after that, the idea was to identify the strands. And for doing that, we used molecular techniques and 16S sequencing just to come up with exact identification of the strands. So for each category of biofertilizers, we distinguish 3 groups. A group of products where we got less strands than expected. A group of products where we got many strands as expected. And the group of products where we got more strands than expected. And you can see that if you check for rhizobia, you have at least 50% of these products where we got more strands than expected. This percentage is much higher with free and fixing bacteria, 70%. And for other PGPR, we have 50%. So it means that most of these products, they contain more strands than expected. So we can suspect some contamination. Just to raise the difficulties because most of these companies, they used to offer you products containing many kinds of microorganisms. But when you have products containing many kinds of microorganisms, you have a lot of difficulties because they used to compete each other's. And it's difficult to get a very good suitable carrier for all the strands. And the N-free fixing bacteria, they have more specific requirements. So sometimes you know that they used to, if the carrier is not good, you can promote contaminants instead of promoting the good strains. So if we go deeply in the results, we distinguish pure cultures, so I mean expected strains with no contaminations. Intermediate products, so some of all expected strains, but with some contaminants, because contaminations can be allowed, but at a certain level, not too high, of course. And you have useless products, so by useless I mean contaminating products, where you have none of the claimed strains, and you have only contaminants. So let's start with rhizobial inoculants. And you can see that only 46% of the strains of the products are pure. But you have 46% of products totally contaminated. I have to say that they come from the same company in Kenya. And when we visited the facilities where these products are produced, we understood why we got these results. People, they use peat that they do not sterilize correctly. The room where they produce the strains is not good. We have got many sources of contamination. So this is the direct impact of this kind of production. The products are completely contaminated. So for free end fixing bacteria, they used to get, the quality of this product used to get lower compared to rhizobial products. And it's exactly what we have got. Almost 43% of the products contaminated here for free end fixing bacteria. And for other PGPR, you can see that it's 20%. So basically, it means that most of the commercial products are contaminated. So the question was to know, because it was a special request made by the foundation, is what is the nature of the contaminants? Do you have an idea? So just to show you an example of list of strains what we came up with. So we have got, of course, Bacillus, Bacillus, so very common bacteria what you can get in the field. Staphylococcus, so it can be dangerous. So we came up with also pathogenic strains, okay, and we got plant pathogens, we got animal pathogens, but we got also human pathogens. So just if I summarize, I'm a farmer. I don't have a lot of money, but I decide to spend one part of my money to buy biofertilizers. And what I've got at the end is I can get sick because I've got human pathogen. So instead of getting more money by getting more yields, I can get sick and I lose my money. Does it make sense? No, of course. It doesn't make sense. But that's the truth. That's the reality. And why do we have this kind of results is, if you check this table now, this is the situation around the world. I was talking about regulations. You have very few countries where you have regulations. France is the worst. Not the worst, but is where the regulation is very strict. If you want to develop products in France, I can tell you that they used to be very strict, and it's very, 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 very important. On the other hand, if you go to US or UK, no regulation about number of rhizobia that you are, or bacteria cells that you are supposed to get in your products, and about the percentage of contaminants that you are allowed to get. So it means that you have a free way for you, if you want to start some business, to produce useless products, snake oil products. You will make money and you won't get blamed. So I mean that if we want to make progress, if we really want to promote biofertilizers, regulation issue is very important. But the decisions are supposed to be taken at the top level. We are just scientists. We don't make any politics. So just to summarize, very low quality of commercial inoculants, presence of pathogenic contaminants, no regulation almost. So it means the main consequence is that farmers lose confidence with biofertilizers. And quality control is very, very important. So all these products will be published in book, which is on the press. And this is a review paper what we published last year, just to describe a little bit all these issues regarding production of biofertilizers and quality. Okay, so just now to discuss a little bit more about formulation of commercial inoculants. This is a work done by a master's student. So the objective was to start from commercial inoculants manufactured by an American company called Baker Underwood in US, and to see if combinations of two strains of Bradyrhizobia combine with or not with Bacillus subtilis strains, PGPR, and to use commercial formulation and to do the same with liquid culture, what we produce by ourselves, and to test it on soybean yields under greenhouse condition. And what we came up with is the fact that whatever was the inoculants, we got an impact on the growth of soybean if you compare with control. But on the other hand, we didn't get any differences between the inoculants. So it means that for this work, whatever was the formulation, the commercial ones and the one that you can prepare by your own, we always got positive impact of the inoculants on the soybean yields. So this work was published last year in this journal, World Journal of Biotechnology, Microbiology and Biotechnology. So after that, we moved to the field. So during the Compro project, after we selected products under greenhouse condition, we moved to the field. This is an example of demonstration trials what we set up in every agroecological zones where we carried out experiments. So you can see this is one treatment, and you can see same soybean varieties. You can see the impact of the rhizobial inoculants compared to this one. So, for example, in Kenya, we were used to be asked, because we have 2 rainy seasons. So farmers, they came to us and they were asking, do I have to inoculate every season, or can I do it once a year just to save money? So the objective, what we did is we identified effective products, what we inoculated in the field in 2 sites, one in the highlands in central Kenya and one in the western Kenya. Central Kenya, they have almost no history for growing soybean. Western Kenya, they have been growing soybean for a long time. So the idea also was to assess the impact of the inoculants in these 2 situations, because we assume that if you have a long history growing soybean, you may promoting some native rhizobia, and competition can be higher. So what we came up with in terms of results is, first of all, what we have got under greenhouse condition was confirmed in the field. I mean, the inoculants what we tested were capable to perform very well with soybean. So we managed to increase soybean yields. But on the other hand, we didn't get any differences between, in terms of yields, grain yields. We didn't get any differences between the treatments, even if we managed to get better nodulation with some treatments. What we observed also is the fact that if you practice inoculations during the first rainy season, you always have impact of the inoculation during the following rainy seasons. So for farmers, it means that farmer can inoculate once a year his field if he wants to grow soybean during the 2 following seasons. So it's by doing that he can save a lot of money. But we observe that nodulation is not directly linked to grain yields, but I will show you more data later. So after that, across the country, we work with farmer associations. And the objective was to organize field days, just to explain what are biofertilizers and to promote biofertilizers. And also we work with farmer associations, and we wanted farmers to set up by their own very simple inoculation trials. One plot with control, one plot with mycorrhiza, one plot with rhizobia, and one plot with both symbiotes, rhizobia and mycorrhiza. And we did it in central Kenya, western Kenya, and Rift Valley here. The objective was for every agroecological zones to get 50 farmers. We provided everything, seeds, products, but they were in charge to set up the trials. They were in charge to follow up the trials, and we had field technicians who came at the end to assist farmers for harvesting. And the objective was to, in terms of scaling up the products, to have an idea about the positive impact or negative impact of biofertilizers. So first of all, we can say that we managed to, so you have 3 colors for 3 provinces that I was talking about, and you have control, rhizobia, mycorrhiza, rhizobia plus mycorrhiza. So basically, we can see that where we put rhizobia alone or rhizobia with mycorrhiza, the yields was pretty good. But no differences between the treatments for the 3 regions. And it was quite surprising because if you check the nodulations, which used to be a very important parameter if you want to assess the impact of your inoculations, if you check nodulation, you can see that you have a significant impact of the treatments on nodulations, specifically here for Bungoma. And we also managed to show that all the nodules were occupied by the strains that we inoculated. So it means that even if all your nodules are occupied by the strains you inoculated, even if a nodulation was stimulated, your yield is not directly affected by the inoculants. So what does it mean? So it means that, for example, you have other factors playing a role, such as, for example, soil characteristic or something like that. So we did the same with combinations of rhizobia and P-solubilizing microorganisms, and we got, this is just for one region, but we got almost the same results. Positive impact on the nodulations, most of the nodules occupied by the inoculated strains, but no differences between the treatments. So it means that now we have to think of how can I optimize my system, how can I inoculate better, or do I have to associate my rhizobia with other microorganisms to be able at the end to get an impact on the yields. So as I told you, we did the work in 3 countries, and just to illustrate a little bit the difficulty when you try to promote one kind of inoculants, it means that because sometimes the best inoculants is not the same according to the country where you work and according to the agroecological zones where you work. For example, in Nigeria, this is these products, what we call RACA6, which perform very well in northern Nigeria under irrigation condition. But in Kenya, these products, Legume Fix, manufactured in UK, gave the best results in Bondo. It didn't give significant results in Bungoma, in Rift Valley. In Chuka, we came up with 2 other products giving good results. And in Ethiopia, in Jima is southwest, Legume Fix again, and these products produced in Zimbabwe gave good results. And in Pawe, just at the border with Sudan, you can see Legume Fix, and you still have the same strain. So it means that for 3 countries, same plant material. We didn't except for Legume Fix. Otherwise, for the other products, the best inoculants was not the same. So what do we have to do? This is typically the difficulty what we have been facing. So my 2 last slides are about they are aiming to raise some important issues. And for example, I told you that in Kenya we ran some tests to see if farmers needed to inoculate once a year or twice a year for growing soybean. So it's questions about persistency of inoculants, and we started getting some results. How can we explain the high viability of a crop growth response to after inoculation of biofertilizers, which have a factor responsible of that? We, for example, in soybean, we run tests on promiscuous soybean varieties selected by ITA in Nigeria. And we contributed to promote this material, very interesting, in other African countries. But what is going to happen if, for example, we want to do the same with new varieties of other legume species? What will be the consequence for biofertilizers? I was talking about the lack of significant responses after inoculation with mycorrhiza and P-solubilizing bacteria. And for people who are interested, I suggest to read this review paper published in 2011. Very interesting, because it's in your mind, it gives you some explanation and it gives you some suggestions where we have to put our efforts for the future. According to my personal experience, I can say that working with the private sector was interesting, but also was very tough, because these people, they don't share the same interests than me. For these people, the objective is to make money, whatever it costs, whatever are the consequences for the end users. The objective is to make money. And we can see the results, because now they can put on the market products with human pathogen. So this is something very, but we cannot work without the private sector. I mean that the private sector needs to be associated somehow, I don't know, but we need to do it. I already raised the issue about regulation, so I think it's very important if we want to be sure that on the local markets we will get effective products. So quality, as I told you, is very important, but it's linked with regulations. And the last point is about the development of new inoculants. I mentioned this new inoculants for soybean with rhizobia and node factors. You have also something that we have been willing to use is mycorrhiza with other molecules, trigolacton, for example. Very important, and people have started getting good results in Europe with that. So maybe something we need to think about. I think it's finished.